Cognitive psychology is, at heart, thinking about thinking. It's what we call metacognition. And there's many layers to metacognition because our body has to go from impulses to thinking, to preserving, to creating representation of the world. And there's got to be a lot of layers in that. So we're going to address this in terms of layers, and we're starting with a structuralist perspective, which means today's episode is about biology and cell activity. This level of analysis is called cognitive neuroscience. These layers go from emotion and thinking to reflex and instinct, but essentially it's all cell behavior. Now originally they considered the brain to be one thing, just whatever it did it was doing. Then we discovered, thanks to the work of Ramon y Cajal, that it's actually a net. What he did was, well, back in the day they would take slices of brain, put them under a microscope, and then they would stain those slices of brain so that the neurons would actually show up. And what they found was, it's a net, it's a network. And Cajal himself did more, because what he found out was that all the little interconnected net were a bunch of individual cells, whereas before they thought it might be one entire piece. So now we find out that our entire brain, our entire nervous system is based on these individual cells that interconnect, but they don't actually touch. Or rather, neurons do touch, but they touch just enough to keep a synapse open between them. Because in that synapse, there's a soup that tells the next neuron what to do. The idea behind all of these neurons acting as a net independently, but together, is called neural doctrine. So first we'll start at the actual cell body, where this is all taking place. Each neuron, there's several different types, but your basic brain neuron is going to have a cell body where the nucleus is, and it has dendrites that pulls in signal information from other neurons to tell this neuron whether to fire or stop firing. That impulse is then sent into an axon, which is an electrical cord, essentially, which transmits the impulse down to what's called terminal buttons, and those terminal buttons turn the signal back into a neurotransmitter for transmission to the next cell to tell the next cell to either fire or stop firing. Now, around the axon, there's this thing called the myelin sheath. Essentially, an electrical cord, to have a signal transmit through it, needs some kind of shielding or insulation. And without insulation, the signal tends to get lost. Well, this myelin, this fatty layer, is around the axon to keep the signal from escaping as it goes through the neuron. Mostly, though, it's because these neurons can be tiny or they can be as long as 2 feet long, so the amount of insulation is rather important in getting the signal to the other end. At the end of the cell, there's this thing called a synapse. It's a gap. And in this gap, there's this soup, because these cells, these neurons, are releasing neurotransmitters, which are activators for the next cell, into this soup, and they get either picked up or retaken up by the original neuron, depending on whether the signal starts, stops, or needs to continue. There's several different types of neuron. The brain neuron is the most normal type, but there's also a thing called, for example, a sensor neuron, and that would be like your pressure gauge on your fingers or your hearing or your eyes, that actually takes a signal from some other source, from a stimuli, and transmits it into an impulse to be sent to the brain for calibration and recognition. Another thing Cajal realized was that these neurotransmitters came out of cells and went into cells in only one direction. Neurons were not two-way streets, they were one-way streets. And another thing that happens in neurons is that each impulse is exactly the same amount of electrical signal, which means the amount of stimuli does not make the signal stronger or weaker, but it makes it fire either more rapidly, more slowly, or in greater numbers of neurons going to the brain. And we call the signals going to the brain afferent because it's affecting us. And we call the signals going from the brain to the body efferent with an E. Now, as those neurons fire, and they fire at exactly the same amount of electrical power each time, there's a set threshold that a sensor must be able to achieve before the impulse gets sent. We consider this to be a gun analogy, or essentially you'll hear it sometimes as the all or nothing, which means there's a threshold, and if the stimuli reaches that threshold, the neuron will send. If it doesn't reach that threshold, the neuron won't send. And it fires like a gun. Either the bullet's going or it's not. The bullet isn't going to change speed. The bullet isn't going to change power. It's just the fact that whether it goes or not. Now, it could be a whole bunch of bullets going, or a rapid fire, but it's just going to be the same bullet with the same amount of powder. It's just going to be the same speed, same velocity each time. When these neurons are not firing, we call that resting potential. Resting potential essentially means that there's not enough electrical charge difference between the inside of the cell and outside of the cell for anything to actually happen. Now, these neurons tend to be negatively charged both on the inside and outside of the cell. But the inside of the cell during the resting phase is going to be 70 millivolts more negative than outside. So what happens, it pushes 2 potassium out to bring in 3 sodium. And it does that, so it's gaining 1 positive charge every time, positively charged potassium going out at 2, coming in at 3, and it happens so fast and so often that eventually the inside of the cell becomes more positively charged than the outside of the cell, and that electromagnetic impulse, if you ever had 2 magnets together and you've had 2 positives and they just push each other away, essentially that's what's going on down the cell. Once it gets more positively charged, it electromagnetically pushes that impulse down the cell till it gets to the other end where it's transmitted back into a neurotransmitter. So you got a bunch of sodium on the inside, a bunch of potassium on the outside, then when it fires, the floodgates open and all the potassium comes back in so it can start over. And this happens, generally speaking, about 200 times a second during a normal firing rate. So what makes a neuron fire? Well, that depends on the neuron, where it is, and what it does. Because not all neurons fire for the same reason. And this is where you get into the layer system. So for example, if you have a sensor neuron in your finger and it gets pushed, then that sensor neuron is going to start an impulse that goes to the brain. It takes that, turns it into an electrical impulse that which they move into a neurotransmitter, usually acetylcholine, to make the next one fire, which transmits down that axon to the next neuron, which makes that fire until eventually it gets to your brain. Then your brain has to decide what to do with it. And most of these impulses are filtered out before it gets anywhere else. But some of these impulses, if they're important enough, will get layered up into the next layer of cognition. Now, we do have structures of the brain that do specific jobs. For example, like the left side of the brain above the ear is Broca's area for doing outgoing speech. Behind that is Wernicke's area for doing symbolism and incoming speech. But we've found that very few things in the brain are actually very centralized. Most of it happens on a broad spectrum across the brain. The cerebral cortex, the outer 5 layers of the brain where memory are stored, cover the entire outside of the brain. And if it actually needs to make more, it'll just make more surface area for it to store it on by creating more wrinkles. So it's a matter of not where does this happen, but where all does this happen. But neurons tend to be very specialized, at least in the human brain. We have, when we're born, one of the few things that we can do is recognize faces. That's because we have what's called a fusiform facial recognition structure. It's for recognizing faces because we, as human beings, have to be able to recognize faces, and that in itself is one's area of the brain. But in order to survive, in order to not be killed by other humans, we have to be able to, 1, identify the face, if we can remember it, so it goes into memory. Identify whether that person is going to be hostile or aggressive or nice. So then we have to figure out what emotions it's processing, which is another part of the brain. Then we have to figure out, are they lying to us, which is a very complex part of the brain and not everybody's good at it. But these things happen at all different areas of the brain and they all have to come together in a split second to know whether that person approaching you out of the woods in the middle of the night holding a weapon is a friend or a foe. Your ability to live and make babies and have future generations depends on it. And essentially, biologically, that's what we're here for, is to make babies and develop and mutate into a better life form to fill a better niche. So being able to process things quickly, especially other humans' facial intentions, critically important to survival. So how is it that we do this so quickly? Well, the short answer is we make mistakes. Our network is not one line of neuron after another line of neuron. It is a net, and in this net, we have this net because it creates a path for a lot of shortcuts. Those shortcuts come with errors, but generally speaking, our brain is just enough to keep us alive or away from death from the hands of other people. So these errors, while sometimes important, generally speaking, if we don't die from it, then our brain isn't going to try to fix it. Most animals, at least the higher mammals, also have what's called feature detectors. And our brains are wired from birth through our experiences to either recognize or not recognize certain things, which is fascinating in the studies of cats. For example, if you put a cat into a black box when they're going through their sensitive period, which is a period of brain growth for everything, essentially eyes, for vision and eyesight, as soon as the cat's eyes open, the brain starts processing that information and uses the stimuli to actually create a framework inside the brain to put all this information around. Now, if you don't let the cat see anything and put the cat in a black box, for this first, 6 weeks of its life, it will never, when it comes out, be able to see normally because the brain has gone past that sensitive critical period and gone on to other things and just accepted that this is a dark world you don't need to see. It will eventually get better, but it's a very slow process. The studies on this in cats from, I think it's Hubel and Wiesel, what they did was they raised a cat, several of them, they had to do this a lot. They raised a cat in an environment that was all vertical black and white lines. And what they found was once the cat's adapted to these black and white lines as the world, it stops seeing vertical lines. It won't even register a vertical line. If they raise a cat in a world of vertical black and white lines, it won't recognize, it won't even acknowledge horizontal lines. It's fascinating material. I don't suggest you do it at home because there's laws against this kind of thing now, but back in the day, this was amazing discovery, breakthroughs. This is called experience-dependent plasticity, as in, when you're born and you're going through these critical stages especially, but this also happens later, your experience will determine how your brain develops. If you spend 30 years writing comedy, you will actually get better at it because your brain is rewiring itself to get better at comedy. If you drive a truck, you will actually get better at it and gain more professionalism and expertise. Expertise is the better word. In doing whatever you're doing, based on the fact that if you do it enough, your brain will actually rewire itself to do it better, faster, more strategically. Because we know that some neurons will refuse to fire at all unless they're hit with that very specific stimuli from a very specific part of the brain that tells this what to do. They just wait. They wait for that one particular stimuli and the brain needs to process it. And that's called hierarchical processing because in order to process this information, you need a specialized brain cell, so it moves up in the system. You've got this thing called sparse coding, when it's sparse because only a few of them are being triggered because it's such a high level stimuli being processed. Or you got what's called population coding, which is a lot of neurons firing because it's a generalized statement that the brain needs to learn a lot about immediately. But essentially, inside everyone's brain, it's a little bit different. It's like a fingerprint. So yes, we're genetically nearly the same. We got the same brain structures and parts that do the general idea of everything, but each individual stores its memories in a little bit different area so that we can't just, broad spectrum say this is where that's going to be, that's going to be. We can do that generally, but as for like the color blue, where is that stored? And that's going to be stored in a lot of places because you know a lot of different kinds of blue. But if you say minnow, where's that going to be stored? Probably a different place for each person. I'm going to hit a little bit on the structure of the brain that we all have in common because there are some commonalities. We're all human. Essentially the four lobes of the brain. You have the frontal lobe, which does our decision making and our thinking. If you remember, everyone should know the story of Phineas Gage. He had the pipe that go through his head. It went through the frontal executive. He lost his impulse control, started becoming angry easier, having trouble controlling his emotions, making wise decisions. Got fired, joined the circus. Died 10 years later from a seizure because those neural impulses have to go somewhere, and if they don't, they're going to probably start seizing. It's possible. It's likely. You've got the occipital in the back, which does vision processing, so the eye takes its stimulus, sends it all the way to the back, and then back up to the midbrain for coordination. You've got the temporal lobes just inside the ear that mostly do the hearing, and you've got the parietal lobes on the top that have the map of the human neural system of the body. It does processing of touch. Frontal, parietal, temporal, and occipital. Those are your basic four structures of the brain, the lobes. Then you've got speech processing, which tends to be left side in right-handed people. In left-handed people, it really could be anywhere, but you'd need an fMRI to really find it. It's probably going to be on the left side, but you never really know. Forward is going to be about the top of the ear is going to be Broca's area for outgoing speech. We covered this before. Behind that's Wernicke area for internal incoming speech and symbol recognition. You've got the hippocampus, which processes memory, and it puts those memories on the cerebral cortex, which is the outer layer of the brain. Then you've got the amygdala. If I teach you nothing else, amygdala does emotion. And then you've got the old part of the brain that we're not really covering so much in this class is for respiration, heartbeat, and keeping you alive. Also filtering out stimuli that's unimportant or that we consider to be unimportant. Those are your major lobes of the brain and structures of the brain. How do we know they're there? Because these days we can do what's called brain imaging. And the most common is going to be your EEG, and that's where they put the little sensors in the cap around your head and it picks up the electromagnetic signals from your brain because processing neurons actually do create a little electromagnetic field that can be picked up on. So we know what parts of the brain from the outside are are active and what parts aren't, but it's not really very specific, but it is really cheap considering. Then you've got CAT scans and fMRIs, and fMRIs are just really expensive. They magnetically take pictures of where your water molecules are and where they're going and what parts to get more water and oxygen we consider to be those parts, the parts that are being activated. It magnetically lines up water particles, lets them go, and then takes a picture of where they're going. And we're working on all kinds of different things into the future. And as soon as we can do real time, we're going to be making really great strides in progress. But the last thing I need to show you right now is that even when our brain is doing nothing, it's got a mode for doing nothing. It's like a sleep mode on a computer. The brain tends to not ever really shut down. It's always either fixing itself even when we're asleep. And when we're not doing anything, when we're bored, it actually triggers a bunch of neurons, a whole network mode of its own, called the default mode network, where these neurons will actually stop firing when you find something that catches your attention. And then when you lose attention and get bored again, this mode network takes off and starts over. Your brain is always running, it's always moving. Even when we don't think it's moving, it's thinking. Because it's got to keep us alive even when we don't know what's going on. And it doesn't tell us everything because it has to filter out all of this. Even that airplane that went over a while ago. Yeah, I heard it. But I didn't acknowledge it. That's what we call cognitive neuroscience. And there's a lot more to it, but this chapter 2 is going to be, yeah, that's all you need to know.